So I want to start by welcoming everyone to Mason's Defense today. Before I do the introduction and pass this off to Mason, I just wanted to review some of the Thesis Defense Zoom etiquette. So all of the participants, all the audience participants are going to be muted throughout the defense. Don't try to turn on your video or share your screen during the talk. And then there will be an opportunity for questions at the end, just like in a normal defense. So once the speaker has finished their talk, you can use Zoom's raise hand feature to notify the meeting host that you have a question. And yeah, that raise hand feature is located under the participants tab at the top of the Zoom window. So if you have a question, and then you will be unmuted and you can ask your question. So I'm going to see if I can take over really quick and share my screen. So it's with great pleasure that I introduce Mason Cole today, and I'm excited for him to share his research with you. He truly is a graduate student extraordinaire. So Mason has had a love of animals since he was a kid, initially more with dinosaurs and dogs, but combined with his interest in marine life, it's not surprising that he ended up getting a master's in the field of marine vertebrate ecophysiology. So Mason actually started at Moss Landing before I did. He was an intern in the vertebrate ecology lab, and by the time I started, he knew a lot more about what was going on with the stranding network, and he was helping different graduate students with their projects than I did. As an intern, he took the physiological ecology class that I taught. It was the first time I'd ever taught it. And I was really impressed with a proposal he wrote in that class after we just kind of had a brief discussion about project ideas, where he actually wrote the proposal as an intern for that class that ended up being his first chapter. So I'm not going to go into too much details because he'll tell you more about that. But based on his work ethic, kind of his contributions to the lab, his performance in that class, and that excellent proposal, I offered him a position in the lab, and I was really excited he accepted, even after suffering through the first time I'd ever taught physiological ecology. And as many of you guys probably know, I tend to cram a lot of material into classes, and this was particularly bad because I had no idea what would fit in 3 hours. And so the exams were long, I didn't hand out slides for note-taking, and yet despite suffering through this class, he still accepted the offer and joined the lab. It's really been a pleasure to have Mason in the lab. He's developed 2 exciting projects. One, he was collaborating with sleuths to study feeding behavior in sea lions. And again, I'm not going to go into too much detail because he'll tell you more about that. I also had the opportunity to work with Mason out at San Nicolas Island, where we were capturing wild sea lions. And he will talk about some of the data he analyzed from wild sea lions in his second chapter. And then probably what was most impressive of all was how he really mastered MATLAB in order to perform the analyses he's going to be talking about today. And he was always, whenever possible, he would put together talks and posters to present his work, and he would get feedback from different people and incorporate that into his project. But in addition to doing excellent thesis work, he also, I wish I had more photos. He also helped everyone in the lab on their project. I'm horrible about taking photos, but we spent many days out in the field working with elephant seals. We've done recoveries down in San Simeon. Like I said, he came out to the Channel Islands. I think he's helped everyone in the lab with their project. And not just in the field. If they had any questions with MATLAB or lab work, he was always willing to help. And he even ran the stranding program for a few years as a graduate student. So he's been an incredibly valued member and mentor to the students in the vertebrate ecology lab. And in addition to always helping in the field and being great, he always had a smile. He was always just such a positive attitude, willing to have fun. You can see he clearly likes to dress up, but he somehow managed to balance working an almost full-time job, being a valued member of the Moss Landing community, and still kind of having a positive attitude and having fun with his friends. So with that, I will pass this back off to Mason. So he can tell you all about the research that he's been doing over the last 2 years using accelerometers to study the energetics in wild sea lions.
All right. Hello, everybody. Welcome, and thanks for listening to this thesis. Hold on, technical difficulties. Okay, we're good. So to start, I'd like to briefly introduce you to my study species. That would be the California sea lion. The big lovable characters that bark at you, the ones flopped across docks all over California. And the work I'm about to share focuses on two aspects of their foraging ecology, but really the question here is why study this? So first of all, California sea lions are very abundant predators, right? With a population approaching about 300,000 individuals. The foraging range, kind of shown here in cross-hatching, centers around rookeries in California and Baja California, extending from beyond the Baja Peninsula up to the southeastern Alaska area. That's the area outlined on the map. So there's a whole lot of them. They're covering a large area. And they target diverse prey throughout this range, and so they exhibit variable diving behavior and strategies. These both open water and seafloor habitats over a wide range of depth, up to over 500 meters. So in this figure from McCurran et al.'s 2016 paper, we see a variety of diving patterns like those at the top of the screen, accompanying a range of spatial habitat use shown on the bottom. The patterns of what they eat and where they forage, they change based on what's going on in the ocean. So for example, El Nino events often cause oceanic anomalies within sea lions' range. This alters the distribution of some of these sea lions' favorite prey, and when that happens, we see sea lions travel greater distances from rookeries or haul-out sites. They go further offshore, they dive deeper, and they dive for a longer duration. And also in those years, we see signs of energetic struggle and reproductive failure. So here, for example, are yearly pup counts, with El Nino years highlighted in red. And the story that emerges from this and quite a bit of other sea lion work is that sea lions are energetically vulnerable to changes in their distribution of their prey. So if you want to know more about why this might be, we got to look at both sides of sea lions' energy balance. So that's energy gain, which is when and where they eat, and energy expense, which is how much energy is spent to forage. So in this thesis, I try to address key aspects of both feeding and energy expenditure, and I do it separately in 2 chapters. So for each chapter, I'll give you the pertinent background and context, walk you through an abbreviated version of my methods, and present my results and discuss what those results mean. There's a little bar at the top of the talk to indicate where we are, in case math makes you sleepy, for instance. So chapter 1, the title is a little bit of a giveaway here. A head-mounted accelerometry accurately detects prey capture in California sea lions. So why do we want to detect feeding using accelerometers? This real 3D track of a sea lion foraging gives us a bit of a visual hint. Simply put, prey capture is difficult to observe in marine species, especially those that forage far from the prying eyes of researchers. We're not going to get a ton of data just looking down from the surface. Can't just follow animals like sea lions as they search for food, sometimes hundreds of meters underwater. So we need an affordable, medium or long duration method for remotely observing foraging. And if we can verify these accelerometry methods, we'll end up with a ton of data that look like this, with feeding observations that complement diving and movement data. Right? And if we can detect foraging, then we can get at patterns of feeding attempts. So we'll be able to see how these patterns compare with foraging strategy, with certain behaviors across dietary niche. We'll be able to look at what these comparisons tell us about reproductive success and survival. So basically, in short, it's an important basic data set that can help answer bigger questions in the future. But first, we need to be able to reliably detect feeding. So a little introduction to accelerometry with pinnipeds. Accelerometers are generally included as one of the few remote sensors that are included on animal-borne biologgers, like those shown in the photo here of the sea lion. An accelerometer measures the acceleration experienced by the sensor in the biologger, usually sampling at least 16 times a second, that's 16 hertz, along 1 to 3 axes. There's a number of studies that use acceleration data to detect feeding by marine mammals, and there's a few variations of this, but in essence, it's always pretty simple. A signal needs to pass a threshold. So in the study shown here, as with several others, feeding is detected when filtered acceleration along one axis surpasses a threshold. In a couple other studies, feeding is detected when the variance of acceleration data surpasses a threshold, which is quite similar in concept. And some studies have also used triaxial jerk calculated from acceleration to detect feeding. So jerk, shown in the bottom panel here, is the rate of change of acceleration. So it's a measure of how rapidly acceleration changes. So in this study, feeding was detected when the calculated root mean squared triaxial jerk within a window surpassed a threshold. But again, it's a signal surpassing a threshold. So with these methods, the elegance is the simplicity, but these methods don't select for specific signals, just big signals. So that means that there can be a pretty high rate of false positive detections, depending on the behavior of the animal. False positives have only been reported in some studies, but where reported, they're usually pretty high and variable between individuals. So the lower we can get the false positive rates, the more accurate our feeding observations will be. So the overarching objective of this chapter was to validate the use of accelerometers to detect feeding in California sea lions. Overall, this was an open search for whichever signal analysis, anything from published methods to something new, minimized false positive detections while maintaining very high true positive detections. I also looked at the effect of tag placement. I suspected that an accelerometer mounted on a sea lion's head is more likely to provide useful data, but that on the other hand, if data from back-mounted tags can detect feeding too, this would be a practically useful result. And looking at the effect of sampling rate, or how often the accelerometer took measurements, I expected that detection accuracy would improve with higher sampling rate due to better signal resolution. Finally, prey size. In this study, I predicted that we'd see correlation between signals of feeding detection and prey length, due both to larger magnitude of acceleration signals and to the longer duration of detected signals. So for all of these points, the goal was really to get practically useful information to aid future studies with wild sea lions. So jumping into the methods, for this chapter, I worked with sleuths just down the road here from MLML. I developed these homemade neoprene head straps in blue after some trial and error, and sleuths trained 2 California sea lions, Callie and Nemo, both adults, one male, one female, to wear these head straps and follow the experimental protocol, which I'll go over in a sec. So I set up 2 GoPro cameras facing different angles at the feeding station and wore one on my head. And the sea lion subject, either Nemo or Callie, stations at the trainer wearing the accelerometer. When the sea lion is sent to the feeding station, a dead herring of known length is released in front of the cameras, which the sea lion then captures and consumes and returns to the trainer for more trials. I performed video analysis before looking at this accelerometry data for those trials. And in short, this involved recording the timestamps of important moments in feeding kinematics. I would take a video like this and the one before it, frame by frame, mark down the timing of when the tag first submerges underwater, when the sea lion's mouth first opens, any other rapid head movements such as a head retraction with suction feeding, a jaw closure, any side-to-side shaking motions, etc. And like I mentioned before, I had 3 cameras that together captured the entirety of each trial. The reason it's getting cut off here is because I just chose one to look good for the camera. For the back-mounted tag location, I went through the same process, but with the accelerometer mounted to the tight-fitting harnesses the sea lions already had. So here's an overview of the analytical design for my accelerometry data analysis. For each sea lion at each tag location, we ran dozens of both feeding trials and control trials. The videos just shown depicted feeding trials. Control trials, the sea lions would swim the same path as in the feeding trials, but would instead be called back to the trainer upon nearing a target without catching prey. So this mimicked the swimming pattern, but without the prey capture motion. A random subset of both trial types was pulled out into a training data set to help inform and train a prey capture detector. The majority of feeding and control trials were reserved for the test data sets on which the independently developed detector was quantitatively tested. Results from the feeding trial test data sets gave true and false positive detection rates and were used in statistical analyses against prey size, while results from the control trial test data set provided information on false positive detections that occurred during the control trials. The training data sets had about 1/3 of the total control and feeding trials for each sea lion. Using the feeding motions from video analysis as a guide, I came up with a wide range of signal requirements that could be helpful in identifying prey capture. I then iteratively tested the full breadth of combinations or phrases of these signal requirements to determine which signal phrases most accurately detected prey capture and minimized false positives in the training data set. So this is what the typical raw acceleration signals of a prey capture trial look like. The sway axis, it's in teal, gives side-to-side acceleration relative to forward swimming. The surge axis in orange gives forward-backward acceleration. And the heave axis in blue gives up and down acceleration. The plot shows you how measured acceleration along these axes changes over time, which you may notice is a really short time scale. I'm going to try to keep this color scheme for these acceleration axes throughout this half of the talk. Besides raw acceleration on the top, there are many other data forms that could be used in signal analysis. So I'm showing here, for example, the triaxial jerk in the middle and the jerk calculated for each axis below that. And really with all this, the question I'm trying to answer is what pattern or combination of patterns is a reliable indicator of feeding? So I'm looking through these signals and others for patterns that are always there during feeding and not there at other points in time. So in other words, what would it be about the signals of the feeding event in the left column, but not in these other two columns, for example, which were not feeding events, that reliably indicates prey capture? You don't need to study these plots. This is just to illustrate the mental process. That's kind of what I was thinking about. Right, and so this is it. This is the general pattern phrase that I found to most reliably indicate prey capture in the training data sets. I work for both sea lions. It's general enough to detect the wide range of prey capture signal patterns and specific enough to not detect other signals. Its basic components are related to the fine scale kinematics of feeding. So first, labeled A, is a sharp, strong heave axis jerk signal. The signal results from the head rapidly tilting back as the jaw opens. You can kind of see that in frame A at the bottom. Next, and almost at the same time as A, is in frame B, a strong deceleration along the swimming surge axis. This is a really quick retraction of the sea lion's head, kind of a halting of forward motion to allow for suction feeding. And that is followed within a fraction of a second by a forward acceleration as the jaw closes and the skull rocks ventrally forward, labeled C. So here we have the heave axis jerk in blue and the surge axis acceleration in orange for all 12 feeding trials in one session at Sleuths. This is what this was the two variables I was looking at with the detector. We can see some variability between each of these 12 trials, but each trial had that initial heave axis spike and the surge axis deceleration and the subsequent surge axis acceleration. And the tricky part here was to write a detection program that's broad enough to detect all of these, but specific enough to not detect other movements, other random things that a sea lion does. And this is an overview of how the back-mounted accelerometer results went for all the trials. Basically, there just wasn't any accelerometer signal at the moment of prey capture at all. Prey capture is shown by the orange rectangle there in the middle of the screen. The x-axis is time in seconds with the 3 axes of raw acceleration on the top panel and triaxial jerk on the bottom panel, just to show you a couple different variables. So what I learned from the back-mounted accelerometer trials is that the specific prey capture motions of the head and neck don't show up in back-mounted accelerometer data. Because of that, I didn't include back-mounted accelerometry data in any of the further analyses. So now we're just thinking about head-mounted accelerometer data. So back to head-mounted accelerometry, because the value of heave axis jerk, that was the blue line in that detector, varies as a function of sampling rate, it was necessary to have the heave axis jerk threshold vary as a function of sampling rate too. So I empirically determined the most accurate threshold for each sampling rate in both sea lions using the training data sets. And luckily, as you can see here, those thresholds varied as a nearly perfect power function with sampling rate. I then incorporated these power curves into the automated detectors. Once developed, I took the automated detectors for Callie and Nemo and tested them blindly and quantitatively on the test data set, which comprised the underwater portion of all the trials that were not included in the training data set. I evaluated true positive and false positive detection rates for each sea lion at each sampling rate, and I investigated relationships between detection data and prey length. I'll explain all of this a bit more. In this study, true positive detection rate was defined as the number of true positive detections divided by the number of actual feeding events, how many of the real events were really detected. False positive detection rate was the number of false positives over the number of actual prey captures, and control false positive detection was the number of detections that occurred during control trials, which were false positives by default because there weren't any fish. The prey used in feeding trials were whole, thawed herring and capelin. The total length of each fish was measured, and feeding order was unrelated to prey length. I used linear regressions to examine relationships between prey length and certain signal attributes of true positive prey captures. And those parameters are shown here graphically. The event duration is the first one, is the time between the dash lines. Maximum heave axis jerk, another one, is the highest point in the blue line within the event duration. Maximum surge axis deceleration is the most negative value of the orange line within the event duration. The integral of the heave axis jerk is shown in the blue diagonal shading, and the absolute value of the integral of the surge axis acceleration is shown in orange cross-hatching and is a mouthful. All right, the moment you've all been waiting for, round one. So this this plot shows how well my detector blindly performed against the test data set. These are the detection rates on the y-axis against the sampling rate on the x-axis. Circles are true positive detection rates. That's the ones up top. Triangles are control false positive detection rates, or in other words, how many non-feeding control trials were detected. Black shapes are Cali, gray shapes are Nemo, white shapes are the results of using Cali's detector on Nemo's test data set. So this is a crossover test with the white data points. It's important because if we're planning to blindly use this detector with sea lions in the wild in the future, we need to have an idea of how well it works with unknown individuals. Cali and Nemo were quite different in their size, quickness, and visual acuity. So if this crossover worked, it would provide confidence in the future use of a general detector on unknown individuals. So what does this show? First, looking at the black and gray triangles on the bottom, we see very low false positive detection rates across all sampling rates. Check, that's good. And at the top, the black and gray circles reveal very high true positive detection rates at 32 Hertz and above, and decreasing sharply below that. And that's actually also really good as well. The white shapes, again, are the crossover test using Cali's detector on Nemo's data. There we see very low false positive detections, but a different trend in true positive detections, which is the white circles. In that crossover test, we see a peak in accuracy at 50 Hertz, a sharp decrease below that like we saw in black and gray, but also a gradual decrease at higher sampling rates. So towards the right of the graph, the white circles are kind of going downwards. And that's because Nemo didn't produce heave axis jerk signals that were as large as Cali's, and therefore it didn't register as a feeding event to a lot of Cali's detector. Cali's detector didn't register it as a feeding event oftentimes. So for comparison, here are the results using the triaxial root mean squared method, which was introduced in a Harper seal study. Here again, I have Cali in black and Nemo in gray. Circles again are true positive rates, and triangles are control false positive rates like the last slide. Squares are false positive feeding detections during feeding trials, which is something I didn't show you on the last slide because there weren't any. But what that means is something like a flipper stroke or a quick rotation or a turn falsely triggered a detection at some point before or after feeding truly occurred. The trends here are a bit reversed from the previous slide. True positive detection rates stay high across all sampling rates, but instead false positive detection rates of both types increase substantially below 50 Hertz. So comparing these two directly, the more complex detector that I developed in this study produced low control false positives and zero feeding trial false positives across the board, with true positive rates really high at 32 Hertz and above and dropping really precipitously below that. The root mean squared triaxial jerk method, which is a bit simpler, does a great job of detecting feeding across the board, but does show moderate control false positive detection rates, which increase quite a bit at lower sampling rates. So looking at the effective sampling rate, this is why true positive detection accuracy varied so strongly at lower sampling rates in the detector I developed. What you're looking at is data from the same feeding trial example that I showed you earlier to describe the components of the detector, but here we're looking at how that signal attenuates with decreasing sampling rates. So in both panels, the signal is red at the highest sampling rates and becomes cooler towards blue as sampling rate decreases to 16 Hertz. The top panel is showing that although some details lost at lower sampling rates, the overall trend is conserved in the acceleration data. But on the bottom, with heave axis jerk, we see a different story. We're seeing a severe loss of signal resolution and magnitude, especially at 32 Hertz and below, to the point where the blue no longer resembles the red. Switching gears, these are the relationships between prey length and detection data for both animals across all sampling rates. Stuff that's bolded is significant. Cali's on the top half, Nemo's on the bottom half. And unfortunately, what really stands out here is that the results were very different among individuals. Cali's data shows promising relationships, but Nemo's generally didn't. And at the end of the day, I think we need more data from more individuals to really answer this, whether or not there's a reliable relationship here. For the sake of time, I'm going to move on, but I'd be happy to answer questions about this later. So the big conclusions from this chapter are, first, the detector succeeded in detecting real feeding and in decreasing false positives relative to published results. Second, this was true for head-mounted acceleration data, but not for back-mounted acceleration data. Third, a sampling rate of at least 32 Hertz was needed for good accuracy, and 50 Hertz seemed like a sweet spot for the use of this detector on multiple individuals, unknown individuals. And finally, there is promise that acceleration data can help infer size, prey size, but this is still inconclusive because of the variation between Cali and Nemo. So I'm going to take a quick sip of water. Okay, chapter 2. Energetic consequences of dive depth revealed with fine scale analyses in California sea lions. A little bit more cryptic about the results. So to start off with, foraging is one of the most energetically expensive behaviors for many predators. This is partially clear just from the distances that some of these animals travel to get to good foraging grounds. So these are tracks of adult female California sea lions who are on land at rookeries to nurse pups, and they head to sea to forage, and then they have to come back to nurse the pup again, and they do this repeatedly for months. And for breath hold divers like sea lions, it's not just distance. Much of their energy expense can come from the dives themselves vertically. So descending underwater to a targeted prey zone, then ascending again. So here's an example from my data, which is nonstop 12-hour foraging bout with many dives between 300 and 400 meters depth, all while traversing a chunk of the ocean like we saw on the last slide. And because foraging is such a large chunk of the sea lion's energy expenditure, disruption to normal patterns has energetic consequences. So what's been seen is that changes in prey distribution leads to different foraging destinations, deeper and longer dives, early termination of lactation, and reproductive failure. Now, in order to evaluate variation in energetic costs of different dive strategies, we need a way to measure energy expenditure at relatively fine timescales in freely foraging animals. There are a couple commonly used proxies for energy expenditure that could be applied, and these rely on accelerometer data. So what you're seeing here is minimum specific acceleration from my data, which is one of a few ways to estimate the overall non-gravitational body acceleration of an animal. So that would be this in blue. Another similar method is stroke rate, just using the frequency of flipper strokes to get a relative proxy of muscular work. These proxies are elegant in theory, but haven't been validated at really fine timescales, less than a couple minutes. So nonetheless, they're used pretty frequently and they're really easy to do. But there's another option to calculate energy expenditure at fine timescales, and that's bioenergetic modeling. So really what this is using physics to estimate how much energy an animal expends to produce the observed motion. So as a sea lion ascends, we can measure its speed, ascent angle, and acceleration using biologgers. And if we know several other variables, then we can calculate the drag and buoyancy forces acting on the sea lion at any point in the dive. Now, bioenergetic modeling with tag data has been used in free-ranging animals in the last 2 decades by a couple research groups at least, but the focus of these studies hasn't really been on energy expenditure. It's more been focused on some hard to estimate variables like body density, coefficient of friction, lung air volume during dives, etc. But there's no reason why we can't use that approach and then take it a step further with sea lions to focus on how much energy is being expended at fine timescales on the order of a few seconds. So to that end, I had 2 goals for this chapter of my thesis. First, to describe fine scale diving energy expenditure during ascents and descents, and along with that to estimate changes in lung volume across depth and estimate tissue density of the body. And secondly, to determine how dive depth affects mean energy expenditure in wild sea lions. So looking at the effects of buoyancy and behavior. A bit about the logic behind the first goal. Sea lions commonly dive to significant depth, often past 100 meters and sometimes past 500 meters, as I mentioned a couple times. As they descend, airspace in the lungs and respiratory system compresses under hydrostatic pressure, like the CT scan of the dolphin lungs you're seeing on the right. And this airspace compression is roughly expected to follow Boyle's law. Therefore, if a sea lion dives with the profile of the blue line on the left of this page, showing depth over time, then the sea lion's buoyancy in the surrounding water would decrease with depth, following a trend like that depicted with the orange line in the middle of the page. And not surprisingly, that shift in buoyancy with depth is reflected in swimming patterns. So here you see time on the x-axis, depth shown in the black line with descent on the left and ascent on the right. And the orange line is minimum specific acceleration, like I showed a few slides ago. And really what it's showing here is the timing and intensity of flipper strokes, which are all those tall, narrow peaks in the orange data. So in descent, we see an initial push to descend to a point of negative buoyancy, then a long glide to depth. And during ascent, the sea lion has to pay energetically for that free ride it took to depth until eventually it can glide back up near the surface. So I put forth a pretty safe hypothesis for this goal, which is that swimming thrust and energy expenditure or power will closely follow patterns of buoyancy during descent and ascent. So in other words, sea lions will exhibit locomotion behavior that saves energy when possible, like gliding to depth. So if a sea lion's buoyancy looks like this, with neutral buoyancy occurring here, I'd expect energy expenditure per unit time to look something like this. A sharp but lessening decrease in power output in red from left to right as the sea lion descends, and then pretty much the opposite trend during ascent. Descent is on the left side of that plot, ascent is on the right side of that plot. A bit about the second goal, which again is to determine how dive depth affects mean energy expenditure for that dive in a sea lion. Here I've rearranged the curves from the previous slide to explain the concept visually. If a sea lion makes a relatively shallow dive, then it will expend a fair amount of energy during descent and relatively less on the way back up. In a deeper dive, the sea lion spends relatively less during descent, but works a lot harder on the way back up. And the question is, in the wild, which dives, shallow or deep, are more expensive per unit time and which are more expensive per unit distance? So I hypothesized that for a given dive, mean power, which is energy per second, and mean cost of transport, which is energy per meter, will increase with dive depth, meaning I expected deeper dives to be more costly per second and per meter. This was based on a few assumptions, including a fair bit of literature. So firstly, that sea lions will behaviorally minimize travel costs by swimming at an optimal speed. It's hard to know ahead of time how fast sea lions will swim, but I thought it was pretty safe to assume that they would generally behave in such a way that minimized cost. Secondly, I assumed from the literature that mean buoyancy affects round-trip travel costs, that is swimming against buoyancy takes more extra energy than is saved by gliding with buoyancy. So mean cost is minimized in dives to a depth where buoyancy averages 0 and increases as dives become shallower or deeper. That's a gross oversimplification, but the point is valid. And finally, I assumed sea lions would have strong negative tissue density, meaning that not all that much fat, more muscle, which would make neutral buoyancy relatively shallow and thus require more energy per second and meter in deeper and deeper and deeper dives. So for this chapter, I analyzed data from a total of 8 individuals collected during 2 previous field efforts, one in 2014 and one in 2012. Both used lactating adult female California sea lions. Both took place at San Nicolas Island and the Channel Islands during the month of November. Mass and maximum girth were collected after capture. The duration of tag deployment ranged from 9 to 16 days. 2014 loggerhead instruments open tags recorded acceleration at 50 hertz and pressure and temperature at 10 hertz, and the 2012 wildlife computers daily diary tags recorded acceleration at 16 hertz and depth at 1 hertz. So getting into my analyses, we'll start with an equation that most of us should recognize. Force equals mass times acceleration. In the case of a diving sea lion, the forces acting on the sea lion are the drag force, F sub d, and the buoyant force, F sub d sine theta. And remember, the buoyant force acting on the sea lion changes as a function of depth. So during initial descent, travel is opposed by strong positive buoyancy, shown in orange in the arrow, which depends on lung volume, and drag, which varies with swim speed, and that's shown in the yellow arrow. As lungs compress with depth, buoyancy becomes increasingly negative and eventually overcomes drag, at which point the sea lion glides downward. The sea lion then faces those same buoyancy forces in the opposite direction upon ascent alongside drag. So that's what we're working with. Based on a number of previous studies, I calculated drag and buoyancy like so. And those plug into the force balance equation here. There will be a test on all this later. That whole thing can then get rearranged to solve for sea lion density, which is the main unknown variable here, without needing a priori knowledge of buoyancy or drag. So now in order to get to a point where I can calculate buoyancy forces, I needed to estimate body density as a function of depth. The objective here was to calculate body density in 5-second intervals, as was done in previous studies, so that we end up with a fine scale picture of how density changes with depth. To do this, we need to figure out speed, acceleration, and descent angle at fine timescales. Using depth from a pressure sensor and body orientation in the water from an accelerometer, I calculated glide speed as change in depth over change in time. And here I'm skipping a whole part where I wrote MATLAB script to identify when sea lions were gliding instead of swimming. But by only using data from glides, we can be reasonably sure that the changes in speed are due to buoyancy changes. So what you're seeing here is all the 5-second intervals of glide speed data during descent for one sea lion. So to get acceleration from glide speed for each sea lion and have it be reliable data, I fit a curve to these glide speed versus depth data, and that curve is shown on the right there. Then using that curve, calculated acceleration in each 5-second window as change in speed over change in time. And one sea lion's fine scale density data for all 5-second glide intervals ends up looking like this in the black circles. So that's kind of like the density data that I would get from gliding. So for comparison, all the seawater density data calculated from the full tag data is shown in red. So the sea lion density data makes sense at first glance. They start pretty much right after the sea lion density surpasses seawater density, which is required for gliding. But that means that we're missing approximately the top 30 meters of density data, and this is where it changes the most. So hence it's important to estimate what's going on with body density in that depth zone too. And to do this, I came up with a simple model to describe each sea lion's body density as a function of depth. This model used a few input parameters. So density is simply mass over volume. For a diving sea lion, volume has two components, the body tissues and the air spaces in the body. And within that equation, tissue volume can be described as body mass over tissue density, which is shown in the bottom equation. And estimating airspace compression with depth, which is the middle term shown here, was a little more complicated. To do this, I followed published equations that describe lung air volume. And then I introduced the term alpha into that equation in this study in order to account for the fact that the structure of sea lion airways are variable and probably less compliant than the unhindered air compression expected by Boyle's law alone. So alpha is the observed rate of respiratory air compression relative to that predicted by Boyle's law. So both diving lung volume, which is the volume of air at the start of the dive in the lungs, and alpha, alongside tissue density, were estimated via iterative model runs against the data, choosing the best model. All right, and here are the body density calculations and model results visually. This chapter has a few sections, so I'm going to present the results and discussion of each section right after the associated methods. So here you see body density data and models as a function of depth for all 8 sea lions in the study. The dots are data from 5-second intervals, and the curved lines are the best fit models applied to those data for each sea lion. The red dotted line is approximately where seawater density falls as a function of depth, just for comparison. A couple things to note. First, all the sea lions share similar trends in their body density to depth. They all have this kind of curve that goes down in the same way. But more strikingly, there's a fair amount of variability among sea lions. And here are the best fit model parameters for each sea lion that go to those models in those lines. And on the right, N is the number of 5-second intervals used in the gliding analysis. Tissue density, that's the third column here, averaged almost 1,060 kilograms per meter cubed, which is denser than other marine mammals for which this analysis has been performed. Mean diving lung volume for each sea lion ranged from 3.5 to 8 liters, which for these individuals works out to about 57 to 100% of estimated total lung capacity. It means they're taking big breaths. And alpha, which is the compression coefficient that I introduced, fit best at 0.3 for all individuals. So that means across the board, respiratory airspace is compressing at 0.3 times the rate expected from Boyle's law. All of this is new information for sea lions. And because there's so much to talk about, I'm just going to discuss results as they're shown, like I said. The first thing here to talk about is that tissue density is quite negatively buoyant, which affects the depth of neutral buoyancy. It also looks like on average, sea lions are diving with pretty full lungs, meaning they're quite buoyant near the surface. And the apparently slow rate of airspace compression relative to Boyle's law indicates variable tissue compliance in sea lions' respiratory anatomy. Basically, there's structures that oppose unhindered air compression. So using those density models for each sea lion, I was then able to estimate energy expenditure. So for each 5-second interval of data during all near vertical ascents and descents, I calculated thrust as the balance of buoyancy and drag forces, following from previous equations, power output as the product of thrust and speed, and power input as power output divided by a conversion factor. That one is also what I refer to as energy expenditure, where the conversion factor is the product of the propeller efficiency and the efficiency by which chemical energy is converted to limb movement. In a flume study by Feldkamp in 1987, really paved the way for a lot of what I did. It showed how propeller efficiency changes as a function of swim speed in California sea lions. So all of these calculations go into getting thrust and energy expenditure at 5-second intervals during descents and ascents. And these are the results of all those calculations. These are the patterns of swimming force and energy expenditure across depth. So here you see mass-specific thrust on the top and power on the bottom during descent on the left and ascent on the right. Different colors refer to different individuals, where purples and blues had highest tissue densities and yellows and greens had the lowest tissue densities. So what you're seeing here is actually the mean trend of all the 5-second interval data for each individual. The mean is the solid line surrounded by a somewhat transparent 95% confidence interval, which for each sea lion is clearly very narrow owing to a very large data set, and that the trends are pretty clear. Thrust and power trends are nearly identical. So from here on out, we're just going to look at power, that's PI, or energy expenditure, because it's a more accurate depiction of real energy expenditure. I didn't run any stats for this objective because every explanatory variable, including depth, was used in the calculation procedure. The main thing to notice in these energy expenditure trends is that buoyancy is the clear driver of the patterns with depth here. So that's what's giving each line its similar curved shape. There's inter-individual variation, but these trends are exactly what we'd expect given buoyancy changes. Also have noticed how density, shown here by line color, appears to drive the mass-specific energy expenditure requirements at a given depth. So that is denser animals are unsurprisingly working harder to ascend against negative buoyancy. That's given the sort of rainbow pattern on the right side there. And really, these graphs show what we'd expected, passive gliding downward below a certain depth. So that's why it's almost 0 on the left side for a majority of those depth ranges. And then working extra hard on the way back up as a result. So you see a lot more work during ascent than descent. If you remember this figure from earlier, which is showing the timing and frequency, frequency and magnitude of flipper strokes, the energy expenditure trends match up nicely with the observed frequency and power of flipper strokes, as represented here by MSA in orange. So now to investigate the effect of dive depth on energy expenditure, I needed to calculate mean thrust and mean PI, that's energy expenditure, for full descents and ascents. And on top of that, at this temporal scale, I could calculate the cost of transport as the total energy expenditure of the total distance traveled. So now, the question, does dive depth affect mean vertical transit costs? This is a complex graph, so we're going to unpack it bit by bit. Each subplot has the data for a different individual sea lion. So there's 8 subplots, each one's a different individual. In each subplot, the x-axis shows dive depth increasing from left to right. The x-axis show either mean thrust, mean power, mean cost of transport for the entire dive. And each data point here was calculated as a 10-meter depth bin plus or minus standard error. So thrust is shown in gray squares, power is shown in empty orange triangles, and cost of transport is shown as filled orange circles. Looking at it like this, we see a bit of variation in trend among individuals, whether they're going up or down from left to right. To parse out these nonlinear trends while allowing for repeated sampling of individuals, I ran generalized additive mixed models, or GAMs, to investigate the effect of dive depth on mean thrust, mean energy expenditure, and mean cost of transport. So as part of that process, the GAMs, that's the stats I ran, fit smoothers to the data, and those smoothers are shown here. And so these are the overall trends, taking all individuals into account for these 3 variables. So thrust on the left here increases with dive depth. Power shows a more complex trend, but also increases with dive depth. And cost of transport, by comparison, decreases with dive depth. So these trends were mostly driven by shallow dives, as you can kind of tell. But the story this is telling is that deep dives are more expensive per unit time. So more energy per second, but less expensive per unit distance. So less energy per meter. This is the corresponding GAM model output. It mostly shows that the fixed effects of dive depth improve the model over a model without dive depth included, so it's important. And that dive depth and differences among individuals explained up to about 71% of the variation in the data. A little bit less in the case of cost of transport. But why might we be seeing these observed trends? So namely, why are thrust and power, which is energy expenditure, increasing on average with depth? But why is cost of transport decreasing on average with depth? Well, one good explanation could be swim speed. In deeper dives, sea lions descended and ascended faster on average than in shallow dives. And increasing swim speed generally corresponded to increasing power. So here the solid arrows are showing mean swim speed in shallow 25-meter dives, with dotted arrows showing mean swim speed in 300-meter dives. So faster mean swim speed, like we see in deeper dives, does qualitatively appear to be consistent with elevated power. And likewise, cost of transport decreases with swim speed and approaches a minimum at swim speeds of about 2.5 meters per second in deeper dives. So it may also make sense that faster swimming in deep dives is contributing to a lower cost of transport. But to make it even more complex, it appears that sea lions are behaviorally responding to swimming against the buoyant force by increasing their speed. So whether ascending or descending, when buoyancy is acting opposite in the direction of swimming, most sea lions increase their speed. That's why you see little upticks on the right side of that graph. This could be coincidence, but it could also be that buoyancy effects are helping drive swim speed, which in turn relates to power and cost of transport trends with depth. There are a couple exceptions on this slide. So most notably, the blue dots on the top are from a deep benthic diver, just swam fast the whole time. And then a shallow benthic diver in yellow just didn't swim fast the whole time. So to recap the hypotheses, for the first hypothesis that energy expenditure will follow buoyancy patterns, this gets a sea lion of approval, and I'm sorry for that pun. And I found a bit of inter-individual difference due to differences in body density and behavior. Hypothesis 2 is that mean energy expenditure and cost of transport will increase with depth. And this gets a sea lion of partial approval. I found that power, which is energy per second, increased with dive depth, but that cost of transport decreased with dive depth. And it looks like behavioral adjustment of swim speed with dive depth and maybe buoyancy may be driving these trends. So I think these results may be pointing to a priority shift along a dive depth gradient. So in shallow dives, where there is a low risk of running out of oxygen, sea lions can swim slowly when not actively chasing prey to decrease their rate of energy expenditure. This is contrasted with deep dives in which sea lions may approach their oxygen store limits. And so in these deep dives, it requires better travel efficiency, meaning a low cost of transport, which is achieved by swimming at an optimal fast speed of around 2.5 meters per second. The side effect of this fast swimming, though, is a faster rate of energy expenditure relative to slower swimming. So deeper dives are more costly for a given time underwater, but does that mean deep diving is a more costly strategy overall? Well, maybe or maybe not. It could be that the extra costs of deep diving are offset by extra nutritional gains, so more or bigger prey, for example. And we know from recent studies, like this one from McKieran et al., that adult female sea lions in this population exhibit individual foraging strategies, including deep, shallow, and any mix of both. And all of these strategies are pretty prevalent in the population. And this prevalence suggests that each strategy is likely successful at an evolutionary scale. Alternatively, deep diving could be a relatively undesirable strategy on a population level. So for the majority of individuals who do not specialize in it, deep diving can be something of a backup plan when shallow prey are scarce. That's the idea. There's some evidence that California sea lions, which is on the left side of each of these graphs, dive shallower and for shorter duration during the productive colder season in the Gulf of California. So when there's a lot of fish and shallow presumably available, they dive shallower because they don't want to dive deeper. And in the California Channel Islands, when prey distribution is shifted during El Nino years, or like the blob, we see a similar trend of deeper and longer duration diving than normal. And this is, of course, accompanied by foraging further offshore, like I talked about, early termination of lactation. And then this leads to the familiar outcome of lots of dead and starving sea lion pups on the beaches. One thing that's unclear, though, is the degree to which the extra cost of deep diving influences this outcome, rather than just sea lions not finding food. So what this thesis chapter brings to the table is evidence that deep diving does require elevated energetic cost while underwater. So at most, the extra cost of deep diving could help drive this trend. and at least deep diving sea lions need to catch more, bigger, or better food to overcome these added costs. So the big outcomes and take homes from this chapter are, number one, I used hydrodynamic glide analysis and some published methods to lay out the first estimates of sea lion body density as a function of depth underwater. And in this process produced estimates of diving lung volume and the rate of respiratory airway compression during natural diving behavior. Number two, sea lions generally adjust their swimming effort based on depth, swimming harder to maintain momentum when buoyancy opposes them and gliding passively to save energy when buoyancy aids them. Most interestingly, I found that the way sea lions behave in the wild is such that deeper dives are more costly per unit time, despite being more efficient on average per meter. So it seems that sea lions are swimming a little bit faster in deep dives as a way to behaviorally maximize their oxygen stores for foraging, for use in hunting during deep dives when oxygen management is crucial. By swimming faster, however, they spend more energy per unit time underwater. And as we just explored, that could be one of a suite of factors that influences energetic and reproductive success. So here's an almost certainly incomplete list of the people and groups that helped me produce this work, helped me learn or improve with something, or supported me in some way. First and foremost, I want to recognize my advisor, Gita McDonald. I honestly couldn't have put her, I could have put her under most of these categories on the screen right now, actually. I have her to thank for the majority of my MLML education, really above and beyond level of feedback in a good way, and for taking what was clearly a personal interest in my success and the success of other students at MLML and in other pursuits. I could ramble on about how awesome Gita is, but suffice to say, I couldn't have asked for a better advisor. Jim Harvey, alongside navigating a scientific institution through a pandemic, found the time to give solid and grounded scientific advice, and I really appreciate that. Colin Ware, alongside great feedback, was really the inspiration behind the concepts I pursued in my second chapter analysis. The Vertebrate Ecology Lab. I love this lab and all the people that have come through it since I've been here. That pretty much sums it up. Thanks for all the enthusiasm, edits, and friendships. Y'all rock. And you know what? The same thing pretty much goes for MLML as a whole. It takes everyone there, both in the spotlight and behind the scenes, to make things work. A particular shout out to Stephanie Flora and Karen Forney, who went to battle with me on some MATLAB puzzles and are pretty much the reason I can code things at all. Sleuths, both the people and the sea lions, were the reason for my first chapter being possible at all. So Jennifer Zelig, Stephanie Scroven, all the whole crew, they went out of their way to collaborate with me on this project, and I really, really appreciate all of their work. A lot of work went into collecting the data that I analyzed in my second chapter, and so I want to thank the people listed here for sharing those data with me, and all the field crews who helped collect those data as well, who is just too many to list. And I knew coming into this program that I'd be mostly supporting myself. I wouldn't have been able to fit this master's degree into my life if it wasn't for the scholarships, grants, and fellowships listed on the bottom right, the jobs listed on the bottom left, a couple scholarships from the family, a bit of lab money, and the good old California government state university grants. And family and friends, what can I say? You make the world go round, and I love you all. And I especially love these two. I'm not about to get all sappy on the internet, but Sloan and Gus, that's the dog on the right, they make me happy. And with that, I'm happy to take any questions while I might or might not sip on a celebratory beverage. And I don't know how to take questions, so I'm waiting for that to happen.
Well, this is Gita. I wish I could think of one right now, but I feel like I've been pestering you for a few years with all of the questions, and I think you did a very nice job of explaining it. I don't see any hand. Oh, I see a hand. Stephanie. I was like, I see some clapping. So I can unmute Stephanie so she can ask the question for you.
Yeah, totally. Thank you.
I muted her again. My computer's being really slow.
That's all right.
Can you hear me?
I can hear you.
Hey, congratulations, dude. Like, so much hard work.
Thank you.
My question's a programming question, as you might imagine. What did you learn most about programming and how it applies to science? Like, what was your big takeaway just from using programming to do a scientific, big picture?
Oh my goodness, that's a tough question. How powerful it is, how awesome it is. Like once you know how to do it, how much of my thesis it delivered for me. God, I mean, that's really the main thing is I didn't know anything about like even really what it was before I started doing it. And also kind of a way of thinking about things in a very logical manner. I find myself thinking about science questions now, with experimental design in mind, but also with how am I going to code this in mind? And is this possible to do in mind? And God, I'd probably think of a lot better answer if I wasn't like, just delivered a defense, but.
That was awesome, dude.
Thank you.
I don't know how to mute myself again. I found it.
So should I click on people or should somebody else click on people? I don't know how to.
Why don't you click on people? Because you can see them now.
Yeah, okay, I finally got my little cursor here. Let's see if I
Yep.
can.
If it doesn't work, let me know, but you should be able to click on them and unmute them.
How do I even, I don't know how to do it. Can you do it?
Okay, I'll do it. You
Thanks.
just gave a talk. So next question. Here we go.
Mason, can you hear me?
Yeah, I can hear whoever said that.
This is Dan. That was great defense, and that was some great data. Those are some great data. I got two questions, one for each chapter. So for chapter 1, did you do any ground truthing, or how would you differentiate between successful and unsuccessful prey capture?
So I kind of take that as two different questions. The first, ground truthing, everything was compared against video analysis. And that's how I came up with what the rates of true and false positive were.
Sure. Sorry, I meant for as it pertains to the success of your prey capture.
Yeah, so in terms of successful versus unsuccessful, I didn't really have any instances where sea lions missed and tried to. So that would be something that would be a lot easier to investigate with like a wild study. You put a video camera on the back of a sea lion, put accelerometers on their heads, and with the detector that I developed, for instance, you would run that script, see what it spits out, and compare that to video. But no, I wasn't able to really do any of that. That was sort of the idea behind the control, though, was to make sure that the signals that we were seeing were not just spinning or fast movements or something like that. But no, there wasn't really a way to even look at an unsuccessful prey capture attempt.
Cool. So for chapter 2, I was wondering if either your GAMs or your seal body density models, how would they shift in relation to the sea lion age?
Sea lion age?
Yeah, or just the body size in general. Is this an allometric or isometric sort of relationship between density and the age or the size, total size?
I don't know if a relationship exists. Density is pretty hard to measure, actually, unless you like put them in a tub of water or something. But it changes, body density changes a lot based on how big of a breath the sea lion has. So I'm not currently aware, I think, of relationships between body density and age or like length or anything in California sea lions. Although I may just be forgetting something, but I suspect that in general, the same trends will exist for any sea lion of any density over Yeah, the same trends are going to be there. Basically, you saw how density affected. You remember that rainbow graph of like purple, then blue, then green, then yellow, those curved lines. So you see how density affects how hard they're working. But in terms of what affects a sea lion's density, I would venture to guess that if you were to do this on like a really young sea lion, that would be vastly different from an adult. These are all adults. And I would also venture to guess that diet and body condition might play a bigger role than age or length, for example. But I might be mistaken. It's been a while since I looked at those literature. So that's about as good as I can answer, I think, with that one.
Cool, thank you.
All right, so I just unmuted Sarah for a question.
Hi, Mason. Great job, of course. And my question is going to be related to your first chapter. I'm sure that's a surprise to you. Curious about your differences between the two individuals and whether those were potentially related to different feeding strategies, so biting versus suction.
Yeah, I expected that question from you, Sarah. Yeah, I didn't look definitively at feeding strategy, and that's mostly because they pretty much all did the same thing most of the time. There were interesting preferences for like which direction the sea lion swallowed the fish. So they would like, I think you mentioned you saw something that they would like spin them around in the water and then swallow them sometimes, but it happened so quickly. But I saw them both do that. I think the biggest differences in terms of the data, the quantitative data that I produced, had to do with the difference between the two sea lions in terms of their kind of ability to find fish quickly. So Nemo has cataracts, and it made it hard sometimes for him to be able to like locate the fish because it's not swimming, it's not producing a trail. So I think the majority of the difference I saw between Callie and Nemo had more to do with Nemo kind of moving slower and then stumbling upon the fish to a greater degree than Callie. He was obviously still finding the fish and consuming it quickly, but relative to Callie, who was this very fast, really zippy, like you compare that one to like one of those dogs that like runs through the air and jumps super high and catches the Frisbee. Right. And Nemo's like lumbering around. But in terms of your raptorial versus pierce feeding versus suction feeding question, I think I almost entirely saw pierce feeding, and I think I'm saying that right. That's the one though. It's mostly suction, and then they'll sort of like bite it halfway and then suction again.
Good memory. Okay. And then I was also curious if you could apply basically kind of that method and technique to other species without having to do all of the ground truthing. Do you think that it would work on other sea lions, fur seals, phocids? Would you have to do all of the ground truthing for each species to get it to be able to pull out those signatures?
I know, right? So I suspect that it would be, I think it would be similar, honestly. Like I think that it would be very similar. But because I got complex with things, there are certain sort of time separations that are applied to those data signals that need to be met. And if those sort of time lags or lack of time lags aren't met, then it wouldn't detect feeding. But I still honestly think that any otariad would probably produce something similar unless they have like a known different feeding mechanism, for example. I did see, actually getting back to your first question, I did see that qualitatively smaller fish didn't need to be eaten twice. And so we saw shorter prey capture signals because it didn't take as long to eat them. And I could imagine that depending on the prey that's being targeted, you'd probably want to adjust the magnitude and length of the signals that you're looking at. But I think you can use my sort of the detector I developed as like a baseline and then just adjust the spacing of things that you're looking for.
That's awesome. How cool. I'll follow up with you via email as well.
Yeah, for sure.
All right, the next question is Sean. So,
hey, Mason, Sean.
Hi, Sean.
Hey, congratulations. You did so great. My question is actually about your second chapter, and I was just wondering if maybe you could expand on the implications of your results in terms of conservation and even like population survivorship as prey availability changes with climate change and things like that.
Yeah, well, it's hard to know whether or not what I found here is having a real effect, or if those effects are due to something different, like prey quality or distance away from shore, time away from the pup, or things like that. There's definitely correlations between these population level effects and prey distribution. But now the mechanism between those things, it could be something like I showed where like they're working harder, or it could be something like they're not finding as many fish because they're not where they typically are. So they're wasting all this time and energy looking and just not finding anything. We wouldn't know that unless we find the feeding detection data. Or it could be, just how far they have to go. Maybe the prey are further offshore, which they are. But in terms of what it would mean, say that my study is like the main driver of those things, which I kind of doubt. I think it would be a contributing factor. So say that was like a real contributing factor, I think that what that would basically mean is that sea lions would have to quickly adjust their strategy, which they're good at. One example I can throw at you is in Liz McCurran's study. I think it was the 2018 one, might have been the 2016 one. She basically showed that sea lions, and she looked at the field metabolic rate, which is the average rate of energy expenditure for a given time in the field, and found that sea lions who dive deeper almost all the time, that's the deep diving specialists, they didn't have a really high rate of energy expenditure. Whereas those sea lions that had a mixed strategy, they kind of mixed it up and did some shallow, some deep. Those sea lions, the ones that dove deeper on average, did have a higher energy expenditure. And I think what that might come down to is that the deep diving specialists are just good at it. They don't need to do it as often. They, are going to be able to probably target fish more efficiently, or whatever they're eating at those depths, which tend to be like large little hake, for instance. And I'm sure a lot of other fish that I forgot. But I think that what would end up happening is sea lions would have to get used to a new normal, which is something that we can relate to. And basically at a population, or maybe at like an age group or demographic level, they'd have to sort of just shift what they're doing and hopefully become better at it if their species is going to continue to do well. But that would be my best guess as to what would happen is, these weird El Nino years are kind of shocking, but if we're seeing a new normal, like always changed prey distribution, sea lions are so generalist and so kind of hardy and I would just expect smart that they'd probably be able to adjust at some point. But yeah, that's my ramble.
All right, I should have been muted. Okay, so we'll go to the next question, which I think is Colin.
Can you hear me? I think I'm unmuted now.
Yes, I can hear you, Colin.
Okay, good. Great presentation, great graphics. Excellent. So I was just had a technical question about about the calculation of drag, where you use frontal surface area, and other people in other papers use total body area, and that's what I used. And you probably by now know the literature better than I do. And I'm just wondering if you have any feeling about the trade-offs between the two, because you'd expect, a skinnier animal, if an animal just gets longer, its frontal area is not going to change, obviously, but it's not but its total area is going to change. So that you might get a different individual difference distribution if you'd use the other metric. Do you have any comments on that?
Yeah, so from my experience with what I was doing here, when I plugged in the total surface area that I calculated using a sort of adjusted girths method, where we took girths at the sort of neck and axillary girths and shoulder, and try to make this sort of sea lion shape and apply that over the whole thing. The numbers I was seeing weren't making sense relative to seawater density, for instance, when glides started, that sort of stuff. I applied the frontal method, which I've actually seen more in hydrodynamic gliding studies. I've seen almost entirely frontal in similar studies that have done this with like elephant seals, for instance. Not obviously a drift glide, like where they're just falling leaf downwards, but for similar studies with animals that are actively swimming face forward through the water, this is the type of, it was frontal surface area that I saw more. And so I went with that one.
Okay.
I see your point though, actually, about the length versus sort of frontal width thing. And yeah, I absolutely expect that could be this. So these are of course estimations, and there's room for error in these, but I just got much more believable results using frontal surface area. And I think that boils down to error in the volume estimations using the, I forget the name of the method, but where you take truncated cones, using the truncated cones method for sea lions. I'm sure it works a lot better for elephant seals, which are actually more like a truncated cone. But yeah, I just didn't have good success with that method, unfortunately.
Okay. Yeah, so the other question is this, you found that for deep dives, they were actually somewhat more efficient, but of course a deep dive still takes much longer and therefore is going to use more energy. So do you, you were not, I don't think, looking at all at energy expenditure at the bottom of the dive, so the horizontal kind of part of the dive when they reach the bottom. One might expect that animals that are shallower diving would spend more time, doing assuming you've got a model where you sort of dive, then you start foraging at whatever depth you choose to forage at, and then you come up again. Then you might expect animals with shallower dives to actually spend longer in the dive, in the foraging part of the dive. Is there any evidence for that?
I think so. I would have to look into the literature, but yeah, I think what you're hitting on is another reason why shallow diving is just like so much more beneficial. If you can get good prey in a shallow dive, then of course, yeah, the less time you spend diving, swimming around, the less energy you're going to expend overall. And just to clarify, what I was finding is it's more efficient per distance in deeper dives, but it's actually less efficient per second in deeper dives, and that's because of the effect of buoyancy is worse than it is better. So yeah, overall, I mean, if I was a sea lion, I'd be looking for, big fatty meals in shallow areas, but I'm not a sea lion. So I'm sure there's something that they know that I don't, but yeah, absolutely. The other disadvantage of a deep diving sea lion is that they're spending most of their dive. And I think actually I did look in my data. I didn't present this result, but I think in my data, let me just sort of scroll through my data here and see if I found any, or if I included it here. I don't think I did, no. This is extra stuff that I didn't talk about, of course. I do think that I found a larger proportion of time spent at the bottom in shallower dives, which makes sense, right? A sea lion's gonna spend as much time as possible, or any diving animal is gonna spend as much time as possible, or as is worthwhile, in the foraging zone. And I did find that qualitatively in the data from these sea lions. So yeah, that makes sense. I would expect that sea lions in shallower dives to generally have a bigger advantage from that perspective. But again, you never know what kind of like jackpots the sea lions are hitting at the depth. Maybe it's worth it.
Yeah, well, presumably it is worth it for some of them, and there are bigger fish down there.
Yeah.
Okay, thanks.
Thank you.
So I think we have one more question and then I'll wrap things up, but I'll unmute Kristen.
Hey, Mason, can you hear me?
I can hear you.
Awesome. Great job today. That was really cool seeing all the results from what you've been working on over the years. I have a question for you about your first chapter, and I was just curious if you guys did any trials with live fish, and if you didn't, if you would expect like the feeding behavior model to be significantly different if the fish are actually kind of moving around.
Yeah, so we looked into that seriously, and we were actually kind of on the train of going to start doing it, and we didn't end up doing it, and I forget the details of why not, but yes, I would expect differences, but from the literature, the differences that are seen is that the signals are bigger, and especially if a sea lion's not like didn't quite get it the first time, the signal might be prolonged and longer. Right. But I made the detector in such a way that wouldn't rule out anything. So all you need is that first mouth opening signal, and then the sort of head retraction signal, and then a jaw closing signal. And if you got those things and they're spaced apart right, and they're big enough, then that's going to qualify as a detection event. And there's not like an upper limit on how big those signals would be. So I think that my inclination is to think that they would be detected. But again, this is one of those things that would sort of be like a next step. I don't know, maybe like contact the Marine Mammal Center and just like throw a bunch of these tags on their heads and feed them fish and see what happens in the data. Yeah, I don't know what the answer would be, but I think that my inclination is to think that it would work and that the signals would be bigger because the sea lions are more actively trying to chase it.
Cool, that makes sense. Thanks, Mason.
Thank you.
All right, well, I think that's all the questions, and we should probably also give Mason a little bit of a break. But thank you guys very much for joining. Mason, if you can stop